Okay, in our next section in genetics, we're going to be looking at probability and Punnett squares. And if you want to reference your textbook, this would be Chapter 11, Section 2. So let's go ahead and start into that and talk about probability first. Now, probability is a math term, and of course, probability is the likelihood that an event will occur. It doesn't mean it will occur, but it's likely that that would be the case. Some examples would be, what's the probability that a coin will land heads up in one toss? Well, if we look at a coin, we know there's two outcomes. There's either the side that is heads or the side that's tails. So what's going to be the probability of getting heads up in one toss? Well, let's look at it in three different forms. If we look at it in fractional form, there's a chance that 1 out of 2, because the simple fact that, or a half, because there's two sides to a coin. If we look at it percentage-wise, it's a 50% chance, because one side is heads and one side is tails. And we could look at this as a ratio, as a 1-to-1 ratio. All right, so as far as landing heads up, all right, there's a 50% chance that it's going to happen. Now, you might be thinking to yourself, okay, that's a coin. What about if we could roll a 4, let's say, on a dice? If we take the die and look at it, it has 6 sides. So our probability there, that's right, you guessed it, would be 1 out of 6. All right. Let's take a look at the product rule, because it comes into effect when we start to look at not just one event, but possibilities in multiple cases. All right. The probability that an independent random event, all right, will occur at the same time or in a row, all right, is the product of the individual probabilities. So in other words, we got to multiply all the individual probabilities together in order to figure out if that independent random event will occur at the same time. Now here I want to warn you, whenever you see questions like this, you have to read them carefully because of the simple fact that you have to know what's being asked of you. All right, let's take a look at this example. If you toss a coin 4 times in a row, what is the probability that it'll land heads up each time? So in this instance, all right, we're going to be tossing it 4 times, and we want to know what the probability is that it'll land heads up each time. Okay? Well, we know that for every time, there's a 1 out of 2 chance that it's going to happen, or half a chance, 50%. All right? So, if we take the 4 times and we multiply them together, that's the product rule here, we can see 1 times 1 times 1 times 1 is 1, and of course 2 times 2 times 2 times 2 is 16. So there's a 1 in 16 chance of doing this, tossing a coin 4 times in a row, that it's going to come up heads all 4 times. Okay? Now, I want to add a disclaimer here. All right? Let's say we extend on this question. What would be the probability of it landing heads up a 5th time? That's right. If you guessed 50% chance, you're correct. You don't product that all together because of the simple fact that previous outcomes have no effect on future ones. And that was one independent question. So you can see how questions can be misleading. So you got to be careful. All right. This question said, what's the probability that it will land heads up each time for the 4 flips? Then we multiply them together. My second question was, what's the probability of getting heads the 5th time? Well, it's still 50%, or 1/2. Now, let's take a look at Punnet squares. I know most of you covered this probably in middle school, but it's good to go back here. And basically a Punnet square is a diagram that can be used to show the potential outcomes of a genetic cross. And here we have an example here. Now I want you to take a look here. We've got the male gametes and the female gametes. And remember, if you're thinking where these gametes come from, that's right, you guessed it, it would be meiosis. And this is where we have the segregation of the particular traits here, or the genes per se. And we can cross these on a Punnet square. So if we have male gamete that is dominant and a female gamete that is recessive, all right, homozygous recessive, homozygous dominant here, All right, and of course, remember what we call those. We call those the true breeders. They are going to have an F1 generation, or are hybrids that carry the dominant and the recessive. Okay, but they're going to show the dominant trait. Now, over in this one here, our male gamete and our female gamete are both the hybrids from this F1 generation here. So we cross them to get an F2 generation. We can see that there's a resurgence of the recessive trait that we didn't see in the F1 over here. All right. Now, we have one true breeder, and of course we have two here that are what we call heterozygous. That means they have the dominant and the recessive, and we have a homozygous recessive. So if we looked at the phenotype, or the actual trait being shown here, all right, there would be a 3-to-1 ratio. 3 would show the dominant and 1 would show the recessive. All right. Now, once again, if we look at this, this is just probability. Okay. This is not telling you that out of the, if they had 4 individual plants, this is exactly what it's going to be. You're going to have 3 or 4 complete organisms. If we're looking at plants and we can say this is tall and the little t is short, all right, that all 3 would be tall and 1 would be short. Remember, this is just a probability. You would look at this for each individual organism that was formed. There's a 3-to-1 chance that it'll be tall versus short. So that is something to keep in mind, and that is one of the pitfalls that a lot of students fall into. Let's take a look at genotype and phenotype. Now, these were 2 of your vocab terms. Remember, organisms with the same phenotype have the same physical appearance. That's what phenotype is. It's just physically, you can see this trait, all right? But they may not have the same genotype. Right? Take a look here. We got purple flowers. One is homozygous dominant, and the other one is heterozygous. They're both going to show purple, all right? But the true fact is, just looking at them doesn't tell us what their genotype is. So, if you have an organism with a specific phenotype, but you're not sure what its genotype or genetic makeup is, how do you determine the genotype of an organism with a dominant trait? Well, we would take that organism and we do a test cross. That is where we cross the dominant. In this case, we take the organism, in this case they have green pea pods versus the little recessive G's here for yellow, and we cross it with a known genotype. All right. Remember, in order to be a yellow pea pod in this scenario, you have to carry both recessive. So we know that's what it is. As far as the green ones here, it could be heterozygous or it could be homozygous dominant. So we do a cross, and by what we get from the cross determines, okay, if I end up with some yellow pea pods on the cross here, I know I had a heterozygous individual here. But if I get all green again, I know this would be 2 true breeders. Remember our terminology here. They'd be 100% green. And of course, that would determine for us what the genotype is for that dominant organism. So, Mendel's principle of assortment, he also looked at not just single factor crosses, but 2-factor crosses or dihybrid crosses. And of course, traits sort independently from one another during gamete formation. That means none of them are linked together. They are independent when gametes are formed. So let's take a look at this example here. We've got peas here, and you can either have a nice round pea or a wrinkled one, or it could also be green or yellow. So let's take a look at this in a bigger form for a dihybrid. Now, on this 2-factor cross here, we have a parental generation up here where they're both true breeders. One is dominant for round and yellow, and the other one is recessive for wrinkled and green. Now, we cross those 2. We know every single one. The F1, the hybrids, remember these are true breeders and these are hybrids here, are going to show the dominant traits, but they're heterozygous. They also carry the recessive from the one parent. So now we cross them, and this is how it's set up. All right. Remember, everything sorts independently. So we just take the R's from here and the Y from here, and then you work the FOIL method. RY, big R, big Y, big R, little y. Little r, little y, little r, big Y. All right, and you see how those are sorted out? Same thing here. It's like doing the FOIL method. All right, big R, big Y, big R, little y, little r, big Y, and little r, little y. And then we cross them here. Big R, big Y, big Y, big R, big R, big R, little y. And we can see the results that we get here. And when you do this with two hybrids, you're always going to see, this ratio here, where the resulting phenotypes, all right, we're going to have 9/16 of them, because there are 16 squares here, are going to be dominant. They're going to have the yellow and round. 3/16 of them will have the round but carry the recessive green, and then consequently 3/16 of them will also have the yellow but be wrinkled, and then 1/16, just 1/16 of them are going to have the recessive. So we can see that recessive come back out. But anytime you take two hybrids and in a two-factor cross, these are the ratios that you are going to see. So to wrap this up, let's talk about making predictions. And remember, we got to remember those pitfalls. Probabilities predict the average outcome of a large number of events. Okay, so like I said, with the pea pods or the flowers that we talked about earlier in our examples, if you cross them and you get 4 flowers, let's say here, it's not going to be in that ratio. Okay, remember, it's just going to show us over a large number of events. That's what we should see. But it's not always the case. Remember, probability cannot predict the outcome of an individual event. Okay? Now, if you think about it, if you have brothers and sisters or a large family, all right, and like my brother and my sister both have large families. My brother has 4 girls and my sister had 4 boys. Now, you think about it, every time there's an individual event, there's a 50% chance that you're either going to have a boy or a girl. All right. So remember, probabilities can't predict the outcome of an individual event. All right. Oh, I've had 3 boys, I'm supposed to have a girl now. No, that's not the case. All right. Remember, it's just for an individual event, you just go with what is there. All right. And you really can't predict an individual event. But if we look at a large number of events, usually probability comes in more clear, and the larger the number, the better results we're going to get. Okay, make sure you review this, make sure you take some notes, write down any questions you may have that you need to have clarified, and of course we'll be discussing this in class as we do an activity related to it. Okay, thanks.